So now what we are going to do is we are going to proceed to the next part of our chapter, biological classification. So let's have a quick recap of what we had studied in the previous lecture. So we are introduced to the classification system based on uses, and then Aristotle's classification, and then the two kingdom classification proposed by Linnaeus, and then the five kingdom system of classification which was proposed by Whittaker. Now we are going to introduce some changes in the format of our videos. Now we are going to introduce this split screen system we are going to see. So the benefits of this split screen system is that we can on one side view the PDF of NCERT, on other side we can view the summary of the pages, summary of the notes we have created through NCERT, and then we are including some previous year questions. We are not including all the questions you would think of all the previous year questions, because biological classification chapter is a vast one and it has myriad number of questions. The possibilities of questions are infinite, and in the previous years there were too many questions asked from this chapter. So we are including only those questions which clear a concept, or which explain a topic completely, or in which there are two options which are against each other, or in which students get confused which is right and which is wrong. So we are including only those questions which are conceptual, which will make us conceptually strong. So this PDF is dotted with questions from previous years that will help us strengthen our concepts. So we are going to introduce another new thing, that is a box coding system. It has quite helped me during my preparation. So this boxed coding system is like this, that will create some boxes before the start of each new chapter. We can create boxes to our liking, and the number of times you have studied the chapter, we will darken it. So you have studied it one time, we will darken one box, and so on and so forth. Now we had finished up to this point in the previous lecture. We had finished up to this point. So we'll start with the three domain system of classification today, and the two kingdom system of classification versus five kingdom system of classification today. So the three domain system has been proposed by Carl Weiss, and so the three domain system has been proposed by Carl Weiss. I think I had said O-typo in the previous video, so it is not O-typo, it was Carl Weiss. It doesn't matter whether it was O-typo or whether it is Carl Weiss, because it will not be asked in our exam, because it is not given in NCERT. You remain assured of that it will never be asked in the exam. So what Carl Weiss did was that he created a new domain out of Archaebacteria. This is the classification proposed by this thing. Let me highlight it. I'll use a pointer instead. So what it did was this thing. So there are organisms, and he divided them into three domains: Archaea and bacteria, and then eukarya. Archaea contained a kingdom Archaebacteria. So what he did was he created a kingdom solely out of Archaebacteria. Archaebacteria is a kingdom, and he created a domain out of it and named it Archaea. Monera, then a kingdom Monera in our five kingdom system classification. This Monera, according to Carl Weiss, contained Eubacteria. This thing contained Eubacteria, and he created a domain out of it and called it bacteria. And then the rest kingdoms of Whittaker, Protista, fungi, plantae, animalia, he united them all and put it under the domain eukarya. Thus he created a three domain system. You can see the three domains are Archaea, bacteria, and eukarya. So he created three domains: Archaea, bacteria, and eukarya. And out of these three domains, there were six kingdoms: Archaebacteria, Monera, and the four kingdoms. So a 3-domain system of classification which contained 6 kingdoms. So what it did was he created a new taxon altogether. He created a new taxon altogether and named it domain. And this was a super taxon instead. It lied above kingdom. We know in what Whittaker and what the 2 kingdom system classification by Linnaeus followed was KP College of Girls School. This KP College of Girls School followed by Linnaeus and again by Whittaker. They both followed the system of kingdom, then comes phylum or division in case of plants, division in case of plants, then class, then comes order, family, genus and species, so on. Genus and then species. What this guy did was add an extra taxon called domain. He added an extra taxon called domain. So in this way, he created a 3-domain system that divides the kingdom Monera into 2 domains. You see, the kingdom Monera in Whittaker's classification contained both Archaea and this kingdom Monera of Whittaker contained both these Archaea and bacteria, or rather say eubacteria. So this kingdom Monera of Whittaker contained both Archaea and bacteria. So what Carl Weiss did was he divided his Monera into 2 domains, Archaea and bacteria, leaving the remaining eukaryotic kingdoms. Now these 2 are prokaryotic. So what this man did was that he divided Monera into 2 domains and left the remaining eukaryotic kingdoms in the 3rd domain, that is eukarya, and thereby 6 kingdom classification. Now the question that came from this domain system was asked, in which 5-kingdom system of classification suggested by R.H. Whittaker. In the light of recent classification, this question was asked in AIPMT 2008. Quite not a recent question, but important altogether. In the light of recent classification of living organisms into 3 domains of life, bacteria, archaea and eukarya, which one of the following statements is true about archaea? The first option is archaea completely differ from both prokaryotes and eukaryotes. Archaea completely differ from prokaryotes. Archaea resemble eukarya in all respects, and archaea have some novel features that are absent in other prokaryotes and eukaryotes. Now, when the options are closed, then what we do? We adopt the option elimination technique. So we first read the first option, archaea, what is, and look for peculiarities in options. Archaea completely differ from both prokaryotes and eukaryotes. Does archaea completely differ from both prokaryotes and eukaryotes? No. Archaea only differs in case of its cell wall structure. It only differs in case of its cell wall structure from both prokaryotes and, yes, it has some features that make it different from prokaryotes and eukaryotes, but it does not completely differ from both. Archaea completely, so this option is wrong. Archaea completely differ from prokaryotes. This option is also wrong. Archaea resemble eukarya in all respects. I said in terms of cell wall it differs, so this option is wrong. Archaea have some novel features. Yes, this question is apt. They are absent in other prokaryotes and eukaryotes, so this is our right answer. So this option elimination techniques helps a lot when we are confused between different options. Now this question AIPMT 2004, this question is asked in AIPMT 2014. 5-kingdom system of classification suggested by R.H. Whittaker is not based on what? Look, this is a confusing question in terms the, if we refer NCERT, अगर हम NCERT refer करते हैं तो we see the main criteria for classification used by him, and this question is also based on criteria of classification. 5-kingdom system of classification suggested by R.H. Whittaker is not based on. First option is presence or absence of a well-defined nucleus. Second is mode of reproduction. Third is mode of nutrition, and fourth complexity of body organization. Now you would say all these options are in NCERT. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Now mode of nutrition is there in the option, complexity of body organization is there in the option, presence or absence of a well-defined. Most students take this answer, but it is wrong altogether, because presence or absence of a well-defined nucleus indicates its prokaryotic or eukaryotic origin. Well-defined nucleus is present in case of eukaryotes, but in case of prokaryotes, a nucleoid is present instead of a well-defined nucleus. You know, nucleoid, it is not bound by nuclear membrane, and the genetic material is just laying around inside the cytoplasm. So in prokaryotes, instead of nucleus, a region which contains the genetic material called nucleoid is present. It has no membrane binding it. In eukaryotes, we have a true nucleus. So presence or absence of a well-defined nucleus is quite a feature. Yes, it is included in 5 kingdom system classification. That's why this man Whitaker has divided organisms into prokaryotic and remaining into eukaryotic. This criteria is definitely used. Mode of nutrition definitely used. You can see mode of nutrition is used. Some are autotrophic, some are heterotrophic. This thing was thought about. And complexity of body organization, obviously complexity of body organization, you can see cellular, some are cellular, some are multicellular, loose tissue, others have tissue, organ, and further others have tissue, organ, and organ systems, up to organ systems. But this thing, can you see it anywhere? Mode of nutrition, can you see this in table? Mode of reproduction, can you see it? No, this thing was not used. We include this in criteria for classification. NCERT includes it, but when we look at this question and we look at the options, now what need says you need to choose the most appropriate options out of these. So the most appropriate out of these is mode of reproduction. As far as we are taught, Whitaker's classification, we have not used mode of reproduction, and neither in this table is given mode of reproduction. So we will not consider mode of reproduction as a criteria. So we'll take this option. This question is also confusing, I would agree. Definitely confusing. This question is, it was asked in AIPMT 2014, and many students had taken the first option, presence or absence of a well-defined nucleus, because NCERT does not directly inform about this option, presence or absence of, but it definitely informs about this thing, prokaryotic and eukaryotic. So the answer goes mode of reproduction, although it is given in the criteria, but well, so this is a star mark question. This was quite confusing question asked in 2014. Now moving on to the next slides, we will see whether we have other questions as well. No. Now we will move to the two kingdom versus 5 kingdom comparison. 2 kingdom by Linnaeus and 5 kingdom by Whitaker. We will move to the comparison. So we will study this comparison from notes because it's quite lengthy from the paragraph. NCERT dedicates a whole paragraph to this thing, up to here, up to the next page here. So NCERT dedicates a whole para. We're not going to read the whole para. We are going to study. So this 2 kingdom system proposed by Linnaeus did was it posed shortcomings, because it did not include everything that Whitaker had intended to. It did not include, it only included the presence of cell wall or the absence of cell wall, whether cell wall is present or whether cell wall is absent. This thing by 2 kingdom. So this 2 kingdom poses problems, and the solutions are given by 5 kingdom. We will see one by one what problems are posed by 2 kingdom system classification and what are the provided solutions by 5 kingdom. So shortcomings of the 2 kingdom system. So the 2 kingdom system in case of prokaryotes and eukaryotes, based on cell wall, placed bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. It put all the things that contain cell wall under plants. All the organisms that were discovered till Linnaeus and that contained cell wall under plants. He put all the things under plants, and those that did not have cell wall, all were put under animals. All were put under animals. So this is the first shortcoming. Prokaryotes and eukaryotes. Second shortcoming, unicellular organisms and multicellular organisms. So Chlamydomonas is a unicellular, and Spirogyra is definitely a multicellular organism. You'd like to see a Spirogyra. Some say Spirozyra, but I said from childhood Spirogyra. So let it be Spirogyra. So this thing Spirogyra is multicellular, I know. So this thing Spirogyra is multicellular, and both were placed under algae. You see, unicellular organisms and multicellular organisms both are placed under alga or under plants. Alga is under plants. So he did not care about prokaryotes and eukaryotes. He did not care about unicellular and multicellular. He was not a caring person at all. He did not care about autotrophic and heterotrophic organisms. No differentiation between fungi and green plants. Fungi is heterotrophic and green plants are autotrophic. And also, if he considered cell wall, he only focused on the presence or absence. He did not even consider the structure of cell wall. He was such a conservative person, I think. He did not even focus on the structure of cell wall. He only considered as the all and invincible factor, presence or absence. Whether it is present, if it is present, plants. If it is absent, animals. So he was quite a conservative person. Also different in cell wall composition. Fungi have cell wall composed of chitin, and green plants have cell wall composed of cellulose. And he put both the things because both the things contain cell wall, so he put both these things under plants. Fungi, chitin, and green plants, cellulose. Now the solution proposed by Five Kingdoms system of classification was that, so these are the three shortcomings: prokaryotes and eukaryotes, unicellular organisms and multicellular organisms, and autotrophic and heterotrophic organisms. So the solution by Five Kingdoms system classification is that all prokaryotes now are placed under Monera. He created a different kingdom for all prokaryotes. So nice move, all prokaryotes under Monera, all unicellular eukaryotes under Protista, and now the kingdom Protista contained Chlamydomonas plus Chlamydomonas chlorella, unicellular alga, unicellular plants in Linnaeus classification, and Paramecium amoeba, unicellular animals in the Linnaeus classification. So both these things were united under kingdom Protista. So these were the solutions. Now we have finished up to this point. Now what we'll do is we'll come on kingdoms of Vertical. So first we will study kingdom Monera. So bacteria are the sole members of kingdom Monera. Now we have categorized Monera here in this form of, form of mind map. So this is a mind map. Monera, the sole members are bacteria. They live in extreme habitats such as hot springs, deserts. Now look this chapter. Look, you will feel boring. Obviously feel boring in this chapter because this chapter is boring. There are no concepts in this chapter. You have to memorize things. You have to learn details. You have to learn habitats, things where they are found. We have learned categorizations and memorized tables, etc. Examples, many of them, no concept at all. Concepts are there in chapters like evolution, chapters like molecular basis of inheritance, and they are quite exciting also to study. But this thing is all about rote learning. So we'll grind on and on. Obviously there will be diagrams dotting this notes I've created. So let's see if it becomes interesting. So this thing Monera, this kingdom Monera, and its sole members are bacteria. It lives in extreme habitats. Now we have to rote learn it. Hot springs, deserts, snow, deep oceans. It is found in every place imaginable as well as unimaginable. Obviously in prose. Most abundant microorganisms Most abundant microorganisms, the Monera are most abundant. We have learned from childhood, most abundant microorganisms, a handful of soil, hundreds of these organisms, many live as parasites, both as ecto and endo. Ecto as well as endo. Look how NCERT hides details from us. Look, also what NCERT has up its sleeves, how it hides details. Look this line, bacteria, the sole members of the kingdom Monera, they are the most, and why dissection is more important of the NCERT lines. They do hide details, and then you say, no, it is out of syllabus, it is not given in NCERT. But we have to dissect each and every line of NCERT. Hundreds of bacteria, they also live in extreme habitats. Many of them live in or on other. What is it trying to convey? In or on? What do you mean by in or on organisms? As parasites, if a parasite is in, it means endo. If it is on, it means ecto. So this is NCERT hiding details. We will analyze it as in means endo, on means ecto. So this thing's done. We'll move on to the next page. So the habit of, so first we'll study the habit of Monera. So Monera both autotrophic, Monerans are both autotrophic and heterotrophic. In case of heterotrophics, they are majority of decomposers. They are majorly decomposers. And in case of autotrophs, they are photosynthetic as well as chemosynthetic. And they prepare their food from inorganic substrates. Now you see, this paragraph is, I think, the most vital in this whole chapter. I would say whole chapter, if not whole chapter, then this kingdom Monera. In this kingdom, this para is, I think, the most vital and has the most probability that students will do it wrong. I think. Now we'll analyze how. So this thing, first question, I'll frame questions. So this first question, food from inorganic substrates. Now what they do is that they do Monerans, they frame options like Monerans prepare their food from inorganic substrates, option number 1. Or we frame it in true false way. Monerans obtain their food from organic substrates. What is the right answer? It's inorganic substrates, obviously. They do not prepare their food from organic substrate, because food in itself, prepared by all the organisms, be it Monerans, is primarily, if not glucose, glycogen, then it must be of something organic in nature. जैसे plants form glucose first of all, then sucrose, then store it as starch, and then animals store it as glycogen. Everything is organic. Food is basically organic in nature, be it Monerans or be it of any kingdom. So they prepare their food from inorganic substrates, but the food is basically organic. So this statement is absolutely false, that they prepare food from organic substrates. They prepare food from inorganic substrates. The first question dealt. Now we move on to the second question. Second question comes from this para. Though the bacterial structure, and I have framed it under important lines repeatedly asked in NEET or asked in test series. Though bacterial structure is very simple, they are very complex in behavior. Look, the bacterial structure is inherently very simple. If you would say it has only one layer, or rather say mucilaginous sheath, etc. This cell wall, mucilaginous sheath, matlab 3-layered system. A mucilaginous sheath, then comes, or in some cases it has capsule. We will not discuss capsule because it's not given in NCERT. So this is mucilaginous sheath, then comes cell wall, and then comes cell membrane, this system. And then it does not have organelles, no organelles in Monerans because they are prokaryotes, no nucleus, no complexity, and no, these are not eukaryotes also, they are prokaryotes. So they have inherently very simple structure, because Monerans were first formed evolutionarily. Also they are quite primitive. They were first formed on earth, and then the rest of organisms were formed from their evolution. Monerans evolved, and then other kingdoms came. So obviously their structure is very simple, but they are very complex in behavior. So if they had to survive to the changing climate, look, Monerans have lived, I think evolution chapter deals with this. Monerans lived for the most time on earth. So they have to have to face different kinds of climate. They have to adapt to different kinds of climate. So they have developed very complexly. They are very complexly developed in terms of behavior. Now, what behavior pertains? The behavior pertains to their food habit, and their metabolic diversity, etc. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. So, how most extensive metabolic diversity? Because some bacteria are autotrophic and some are heterotrophic. And among autotrophs, they are not only photosynthetic autotrophic, which plants are. Plants are only photosynthetic autotrophic, or in some cases heterotrophs, where they are insectivorous, or in some cases of parasites such as angiosperms, such as Cascuta. But there are exceptions. Plants are somewhat primarily photosynthetic autotrophic. But monerans, they are autotrophic, they are heterotrophic. Among heterotrophs, they are parasites, and they are decomposers. Among autotrophs, they are chemosynthetic autotrophs and photosynthetic autotrophs. So they are metabolically very much diverse, and this pertains to this feature, complex in behavior. So how they ask questions? So one first question here, they ask from here, food from inorganic substrates. And the second question they ask is, though the bacterial structure, they frame questions like this, they frame options like this: though the bacterial structure is very complex, they are very simple in behavior. They only change words, they only change the placing of the words, they only change, what they do is they only change the placement of the words. Very simple, they put it here. Complex, they put it here. And out comes a new question. Though the bacterial structure is very complex, they are very simple in behavior. Now you would get easily confused. We would tick yes, it is right, right option. We had at certain point of time in our lives, we had one NCERT book, and we had read a page, we had read a chapter, biological classification. I think I have heard this line. Yes, I think, though the bacterial structure, we would not focus on these words. We would definitely not focus on these words. We would think, yes, I have read this question. Yes, I'm quite smart. Though the bacterial structure is very complex, they are very simple in behavior. Obviously, they will be simple in behavior. They are evolutionary, and bacterial structure is quite complex. It appeals to us because bacterial structure, we see there's this glycocalyx, there's this capsule, then cell wall, then these things and those, and yes, it would be quite complex. But this is our mistake. The bacterial structure is very simple, but they are very complex in behavior. This is star mark question, quite asked in test series and altogether in different tests. Now, compared to many other organisms, the second question comes out, the third question, second is this, third question comes out from this this line: compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Now what they do is, compared to many other organisms, bacteria is a group show the least extensive metabolic diversity. We would comply to this easily, to this statement of least extensive. Our common sense will comply to this easily. We would easily believe, yes, it must be least extensive, because bacteria are so evolutionary backward, they must have least extensive metabolic diversity. Our common sense often leads us into ditches. So we'll highlight this part. What we'll do is highlight this part in our NCERT, in our respective NCERT textbooks. Compared to many other organisms, bacteria as a group, so the least extensive metabolic diversity is altogether totally wrong. Compared to many other organisms, bacteria as groups show the most extensive. This most word is quite important, most extensive metabolic diversity. Now the rest of para is not that much of importance. Some of the bacteria are autotrophic. You know they synthesize their food from inorganic substrates. We have discussed this. They may be photosynthetic autotrophic, chemosynthetic autotrophic, vast majority heterotrophs. They depend on other heterotrophs. You know the definition of heterotrophs. They depend on other organisms or on dead organic matter. They are decomposers. Now we come on archaebacteria. First of all, we discuss bacteria based on shapes. So this one para discusses bacteria based on shapes. So bacteria are grouped under 4 categories based on their shape: the spherical coccus, this is coccus, plural cocci, bacillus, plural bacilli, spirillum, plural spirili, and vibrium, plural vibrio. So this is spherical, rod shaped, we can see rod shaped, coma shaped, vibrio, and the spiral spirillum. It is quite a, you just have to memorize things. Now we move on to archaebacteria. So these bacteria are special. These are quite special bacteria, as we are discussing the three domain system. There was that question. It also had pointed out to these bacteria are special. They have some novel features over others. So these bacteria are quite special since they live in some of the most harsh habitats. And why they do live in some, how can they live in these most harsh habitats? Due to their cell walls. They have something in their cell wall. We will not discuss what thing, because it is not given in CRT. It will never be asked. And if it is given only cell wall structure, then need people will only ask for what, how they live in the most harsh habitats. We will answer, we will tick cell wall structure. So they live in some special conditions, most harsh habitats such as extreme salty areas, and then they are called halophiles if they live in hot springs, thermoacidophiles and marshy regions, and methanogens. Methanogens are also found in gut of ruminant animals such as cows and buffaloes, and they are responsible for the production of methane, that is biogas, and biogas is used in many. Biogas is used as fuel in villages, etc. Now the archaebacteria, halophiles, extremely salty area. How would we remember extremely salty areas, halophiles? Now we look halogens. What are halogens? What are halogens? What are these halogens? These halogens are salt producers. And how would you remember it? Halogens. Halo plus gen. Halo means salt. This halo thing means salt, and gen means generator. Salt generator. Not we take a halogen such as chlorine. What does chlorine do? Chlorine, when reacts with a metal, it produces a salt called NaCl, salt, any salt of that salt. Now what this halo thing means, I'd only ascribe this fact because we would like to remember extremely, like to connect this salty with halo thing so that we can memorize it in our memories. Extreme salty areas. Salt means halo. Halo refers to halogens, halogen, and this word halo comes from this word halogen comes from halo. Both have halo in common unit. So this connecting of things often helps. This connection of things often helps in remembering us things. So this halogens means salt generator. So this also contains halo, and this halophiles live in extreme salty areas. It also contains halo. Both have halo, and this halo refers to salt. So this will help in remembering these things. Hot springs, hot, and thermo, quite easy to remember. And MC methanogens and MC marshy areas, it's quite easy to remember. Now this para also contains a quite important thing for questions. I had highlighted it in bold form. Archaebacteria differ from other bacteria in having different cell wall structure, and this feature is responsible for their survival in extreme conditions. This question is quite famous in test series. Okay, why do archaebacteria able to live in extreme conditions? Because they have different cell wall as compared to other bacteria, different cell wall composition in comparison to other bacteria. And now comes eubacteria. Then there are thousands of different eubacteria. U means true. This U thing means true. U means true. U thing means true. So these thousands of different eubacteria, and there are two bacteria that are characterized by presence of rigid cell wall. If motile, they have a flagellum. And then comes photosynthetic. Then comes autotrophic eubacteria and heterotrophic eubacteria. Among autotrophic, we have photosynthetic, and then we have chemosynthetic autotrophic bacteria. And among photosynthetic autotrophs, cyanobacteria form a major part. Cyanobacteria also referred to as blue-green algae, and the peculiarity of cyanobacteria of blue-green algae is that they have chlorophyll A similar to green plants. And the cyanobacteria are unicellular, they are colonial, they are filamentous, they are freshwater marine habitats, terrestrial algae. Colonies generally surrounded by gelatinous seed, often forms bloom in polluted water bodies, are responsible for forming blooms. Some of these organisms can fix atmospheric nitrogen. Specialized cells called heterotrophs, everyone knows it. Nostoc and Anabina are examples. Then comes chemosynthetic autotrophic bacteria. They oxidize various inorganic, it is quite apt to notice this, oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for their ATP production. So what they do is they oxidize nitrates, nitrites, and ammonia. Oxidize, not reduce. This word is very important. And inorganic, not organic. Chemosynthetic autotrophic bacteria do is they oxidize, not reduce. They oxidize various inorganic, not organic. So how will they frame questions from this? Chemosynthetic autotrophic bacteria reduce various inorganic substances. False. Chemosynthetic autotrophic bacteria oxidize various organic substances. False. Chemosynthetic autotrophic bacteria reduce various organic substances. Again false. And chemosynthetic autotrophic bacteria oxidize various inorganic, this time true, substances such as nitrates, nitrites and ammonia, and use the release energy for the ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur, NPFES. So they recycle these nutrients, nitrogen, phosphorus, iron and sulfur. Now these heterotrophic bacteria, they are most abundant in nature. Obviously we have pointed out these things in the previous slide. They are most abundant in nature. This thing tells us that these are abundant in nature, most abundant in nature. The majority are important decomposers. Again, the majority are important decomposers. Many of them significant impact. They are both pros and cons, benefits as well as side effects for humans. Among benefits, they help in making curd from milk. They help in production of antibiotics, fixing nitrogen in legumes, roots. Some are pathogens. And among the side effects, among the diseases they cause are that they cause damage to human beings and crops, farm animals and pets, cholera, typhoid, tetanus, citrus canker, as well as known disease caused by. Now these disease, everyone knows these diseases. Cholera is caused by bacteria, typhoid is caused by bacteria, tetanus is caused by bacteria, citrus canker. People confuse that citrus canker is caused by virus. People get confused that citrus canker is caused by virus, but it is not the case. So what you do is that write in this open space the citrus canker virus wrong, bacteria true. So whenever you would revisit this chapter, so you would know on which point you commit mistakes. So only that point you have to revise, not the whole chapter. So the citrus canker is caused by bacteria, not virus. Bacteria reproduce mainly by fission. Fission is asexual, obviously asexual form of reproduction. Now this para deals with the reproduction in bacteria. This para deals with reproduction in bacteria mainly by fission, sometimes under unfavorable conditions. Unfavorably, they produce spores. Under unfavorable conditions. They also reproduce by a sort of sexual reproduction, not sexual reproduction totally altogether. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria into another. And now in this para they dealt with the mycoplasmas. Mycoplasmas are organisms that completely lack a cell wall. They have not cell wall at all. They're the smallest living cell known. It can survive without oxygen के बिना सर्वाइव कर पाते हैं. Many mycoplasma are pathogenic in animals and plants. Mycoplasmas are pathogenic. Now what we have done is we have categorized these things. Now you see how to form notes. Note making is an art altogether. Now eubacteria means true bacteria. Their characteristic cell wall is rigid, motility, flagellum. They are motile through flagellum, or rather say they locomote through flagellum if flagellum is present. They reproduce in three ways: binary fission, spores and sexually, a sort of sexual reproduction. Then the remarks, the binary fission mainly, spores in unfavorable conditions, this thing is quite star marked, and sexual primitive DNA transfer. Now these types, they are autotrophs, heterotrophs. Autotrophs divided in photosynthetic, chemosynthetic. Photosynthetic have cyanobacteria as the majority, and their habitat is freshwater, marine and terrestrial. Their types are unicellular, colonial, and these notes and materials not from outside but sorted out from the NCERT. Freshwater, marine and terrestrial. Types are unicellular, colonial and filamentous. A speciality that they fix atmospheric nitrogen in heterosis. This heterosis thing is important. Their examples are Nostoc and Inavina. And then comes the chemosynthetic autotrophs. What they do is they oxidize, not reduce. They oxidize inorganic substances, and again inorganic, not organic substances. Such as they oxidize nitrates, nitrites and ammonia, and they create energy out of that, and then they use this energy to form ATP. And then the bulb point, the highlighted point is that these colonies of the cyanobacteria are surrounded by gelatinous seed, and they form water blooms in polluted water bodies. They form water blooms in polluted water bodies. they form water blooms in polluted water bodies, and then they recycle nutrients. They recycle nutrients like N, P, F, E, and S, nitrogen, phosphorus, sulfur, and what they do is recycle nutrients such as nitrogen, phosphorus, iron, and sulfur. And then comes heterotrophs. You have dealt with autotrophs. Autotrophs is being dealt. Then we come to heterotrophs. So these heterotrophs, they are most abundant, and majority of them are decomposers. They have their benefits and their harms respectively. Among benefits, their milk to curd, antibiotics, nitrogen fixation, and nitrogen and legumes and roots. And then their pathogens. They cause damage to human beings, crops, farm animals, and pets. And then they cause cholera, typhoid, and tetanus. Citrus canker is quite important. We have discussed it. They are not caused by viruses. They are caused by bacteria. Now let's see, I like to see what this thing called water bloom. This is water bloom. This is water bloom caused by cyanobacteria. This is what water bloom is. And this is what citrus canker is. Why did not come here? This thing is what citrus canker is. This is what we are seeing. This is what citrus canker is. It's like a cancer of the citrus plant. This is what citrus canker is. It is caused by bacteria, not viruses. Remember that. Now mycoplasmas, they lack mycoplasmas. What they do? These mycoplasma completely lack a cell wall. They are the smallest living cells, can survive without oxygen. They are pathogenic in animals and plants. They are pathogenic in animals and plants. Now we quickly solve a question. Question from this paragraph, this 2 versus 5 comparison. In the 5 kingdom classification, Chlamydomonas and Chlorella have been included. In the 5 kingdom system, Chlamydomonas and Chlorella have been included under Protista. Quite an easy question to deal with. And then the next question, AIPMT 210, asked in AIPMT 210. Select the correct combination of the statement A to D regarding the characteristics of certain organisms. Methanogens are archaebacteria which produce methane in marsh areas, right? Nostoc is filamentous blue-green algae which fixes, right? Chemosynthetic autotrophic bacteria synthesize cellulose from glucose. No. Chemosynthetic autotrophic bacteria synthesize cellulose, not cellulose and not from glucose. Chemosynthetic autotrophic bacteria synthesize glucose from, or synthesize food from inorganic substrates. Mycoplasma lack a cell wall and can survive without oxygen. Mycoplasma lack a cell wall. Yes, they lack a cell wall. They lack a cell wall and can survive without oxygen. Yes, they can survive without oxygen. So these 3 options A, B, and D are right, and this 3rd option is our correct answer. Now we move to the our kingdom Monera is finished. Now we move to another kingdom Protista. All single cell eukaryotes are So unicellular eukaryotes are put under Protista. Unicellular eukaryotes, not multicellular eukaryotes and not multicellular prokaryotes. Only unicellular eukaryotes are placed into. So what they would ask? They would ask that kingdom Protista contains unicellular and multicellular eukaryotes. Unicellular eukaryotes, and then they will ask unicellular plus multicellular eukaryotes, and then they will ask unicellular prokaryotes. Now our smartness will allow us to view this option, tick this option. Why not include everything, unicellular as well as multicellular eukaryotes? We are all inclusive people. We try to put together everything under one roof. We are excited to put everything. So this is not the right answer. Unicellular eukaryote is true, but not multicellular. Multicellular absolutely not under kingdom Protista. Kingdom Protista only contains unicellular, and that too eukaryotic organisms, not prokaryotes. So kingdom Protista has all single cell eukaryotes and they are placed in the Protista, but the boundaries of this kingdom are not well defined. Look, this statement is only used in the case of kingdom Protista. You would come across questions in which they would ask key, like this question, select the correct combination of the statement, and they would say in which kingdom. Protista is the kingdom in which boundaries of this kingdom are not well defined. So this is quite an important line. What may be a photosynthetic protist to one biologist may be a plant to another. For example, in case of Chlamydomonas, it is a photosynthetic protistan, but another biologist may put it under algae, under plants. In this book we include Chrysophytes, Dinoflagellates, Euglenoids, slime molds, and protozoans under protist. Members of protist are primarily aquatic. This is a point to be noted. It is primarily aquatic. You see most of the protistans such as euglenoids and amoeba also we have studied from our childhood, that they are found in water bodies, in stagnant waters, etc. This kingdom forms a link with others dealing with plants, and so this is a linking thing. In evolution chapter it is studied topic that what thing links one to another. So this kingdom forms a link between plant and animal kingdom. So this kingdom forms a link with others dealing with plants, animals, and fungi. This kingdom forms a link with others dealing with plants, animals, and fungi. Being eukaryotes, the protistan cell body contains a well-defined, being eukaryotic also it will definitely contain a well-defined nucleus, not a nucleoid altogether, and other membrane-bound organelles. Prokaryotes do not contain nucleus, and neither they contain membrane-bound organelles. Membrane-bound organelles are not present. Organelles are not present altogether. Neither they contain nucleus, they contain nuclei instead. Being eukaryotes, they have membrane-bound organelles and they have well-defined nucleus. For locomotion, some have flagella, some have cilia, some locomote through pseudopodia also. Protists reproduce asexually and sexually. Both asexual and sexual reproduction is found, and by a process involving cell fusion and zygote formation. So they have zygote formation and cell fusion also. Now what we have done is kingdom Protista, Chrysophytes, and Dinoflagellates, Euglenoids, slime molds, and protozoans. So discussed each of these kingdoms, each of these one by one. So here comes Chrysophytes. So this thing Chrysophyte is divided, also divided into diatoms and desmids. Dinoflagellates are not divided in NCERT. Euglenoids not divided. Slime molds not divided. Protozoans are again divided into amoeboid, flagellated, ciliated, and sporozoans. Chrysophytes, they have their habitat in freshwater as well as marine water. Chrysophytes are microscopic. Chrysophytes have diatoms plus desmids. Desmids are also called golden algae. And they are majorly photosynthetic. Phytoplanktons, you had heard of phytoplanktons. So these are planktons and they float passively. So the answer is mostly asked which float passively. So these are chrysophytes, and then they are protistans. Chrysophytes are protistans. So this group includes diatoms and golden algae, desmids. Golden algae are also called desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, and they are called planktons. Most of them are photosynthetic. In diatoms. Now we will discuss the structure of diatoms. I have got quite a nice picture for it. So diatoms have a soap box like structure. साबुन के बक्से जैसा structures, so box like structure. The diatoms are the chief producers of ocean. They chiefly produce in ocean. Their main contribution of production in oceans is of diatoms, and then they have these box like structures, and again soap box, not merely any box, soap box like structure. So these diatoms have two thin overlapping cells. You see this cell and this cell. This cell is overlapping over this cell. Two thin overlapping cells. These are cell walls. These are the cell walls of diatoms. Cell wall, soap box in pattern, and these are embedded with silica. So they are quite strong, and they do not decompose easily. They do not degrade easily. They have silica embedded in them, and the cell wall structure of diatoms. And one cell, this time cell is overlapping the another cell, and they are embedded with silica and thus indestructible. They are not destroyed easily, and they form deposits. If they are indestructible by natural means, they form deposits, silica cell wall deposits after death. They form deposits after death, and these deposits in the ocean bed, these deposits are called diatomaceous earth. These deposits are called diatomaceous earths, and these are quite useful for human beings. They are used in polishing, filtration of oil and syrups. Now these are quite gritty, and used in polishing, filtration of oils and syrups, and they are the cheap producers. Now we come to dinoflagellates. Mostly marine. Their habitat is mostly marine. Their habit is mostly photosynthetic. Their color is yellow, green, brown, blue, red, depending on the main pigments present in their cells. If the main pigment is in yellow in color, then they are yellow. If the main pigment is green, then the green, brown, blue, and red, and so forth. And they have stiff cellulose plates. On the outer surface. You see this is the outer surface of dinoflagellate. Outer surface, they have stiff cellulose plates. And then for locomotion, they have flagella. One is longitudinal. This is longitudinal flagella, and then there is transverse flagella. Transverse is this. Now this longitudinal and transverse, this is our earth. You have heard in case of earth that this is longitude, this is latitude. So this longitude is along the thing, and latitude is perpendicular to the thing. You would hear in chapters like anatomy that this is the transverse section of plants. This is the transverse. So this is the plant, and this is transverse section. This thing is transverse section. So this is perpendicular thing. Longitudinal section we have like this longitudinal section. So this longitudinal flagella, and this is the transverse, and present in a furrow between these two stiff plates. This is present in a furrow. So very often transversely in a furrow between the wall plates, and very often such a dinoflagellate known as Gonia iulex, it comes under red dinoflagellates. Example, Gonia iulex undergoes such rapid multiplication. They undergo such rapid multiplication that they make the sea appear red and cause red tides. This is the red tide. You can see this thing is red tide. This thing is red tide. And is quite harmful for living beings, quite harmful for marine life. This thing red tide is quite harmful for marine life, and they are quite toxic for fishes. They multiply rapidly. Gonia iulex causes it. Gonia iulex is a type of dinoflagellate and come under red dinoflagellates that are quite harmful for marine animals such as fishes. And they release toxins. These things, Gonia iulex, multiply rapidly, and they release toxins. What they release? They release toxins which make these waters poisonous, and this toxic thing then kills marine animals such as fishes. And now comes the third, euglenoids. Majority of them are found in fresh water, and that too in a stagnant, and their habit is heterotrophic. If look, this night thing, this is night, this is our moon, night. So I only use this picture not to say that in only night they are heterotrophic. No, when sunlight is not present, or when the sunlight is deprived in any way, then they are heterotrophic in habit, and when sunlight is fully present, they are not deprived of sunlight, then they so autotrophic habit. Their speciality is that they do not have cell wall. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible, which provides flexibility. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. This thing is covered by pellicle. They do not have cell wall. This thing is pellicle, and this thing makes their body flexible, provides flexibility. And euglenoids, example, euglena. In locomotion. For locomotion, they have flagella. One is short, this is quite short, and one is long, this is long flagella. So nothing quite important here. Majority of them are freshwater organisms discussed instead of cell wall, the protein. This thing is the most important part, I think, of this para. They have two flagella, short and a long one, though they are photosynthetic. In the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoids are identical to those. This thing is very important. The pigments of euglenoids are identical. In one case also we have discussed, in case of BGA here, BGA. We had discussed that chlorophyll A is similar to green plants. The cyanobacteria, also referred to as blue-green alga, have chlorophyll A is similar to green plants, and this thing written here that the pigments of euglenoids are identical to those present in higher plants. Its example is euglena. Now we come to slime molds. Slime molds are saprophytic protists. So in habit it is saprophytic, and the body moves along decaying twigs, सरते हुए twigs and leaves, सरते हुए पत्ते, engulfing organic material को engulf करता है under suitable conditions. So if the condition is favorable, if the condition is suitable, they form an aggregation called plasmodium. They form an aggregation called plasmodium under favorable conditions, and if the that which may grow and spread over several feet. This plasmodium thing grows and spreads over several feet. This plasmodium thing grows and spreads over several feet during unfavorable conditions. If the conditions are not favorable, the plasmodium differentiates. This plasmodium differentiates and forms fruiting bodies. They form fruiting bodies, and these fruiting bodies bear spores at their tips. Now these things are not quite as important as this thing, that these spores contain true walls, and that's why they are extremely resistant. This thing is very important. This thing is not that much important. They are not that much important as this thing, that these spores are bear true walls, and these true walls are so strong that they make these things extremely resistant, these spores, and they help in the air current dispersal. These spores disperse by air current. The spores disperse by, now how can they form questions? They can form questions that the spores of slime molds disperse through water, they disperse through air, they disperse by insects, and so on. So we would take air, not water, not insects. They disperse through air, so they perform air current dispersal. And then they would ask that the spores of slime molds do not bear walls. They have asked many times in test series that the spores of slime molds do not bear walls, but they bear true walls, and that too not false, true walls they bear, and they are extremely resistant and air current dispersal. This plasmodium grows and spreads over several feet. This is slime mold. This slimy, buttery, sketchy thing is slime mold. Now we would like to discuss a question of AIMS 2006 question.